नमस्कार दोस्तों, वेलकम टू चैनल टॉक्सिकोलॉजी ऑन बोर्ड. In this video, we will be discussing about DABT topic, IND as well as ICH S6 guideline. So directly let's go to the question. The question is: conventional approaches to toxicity testing of pharmaceuticals may not be appropriate for biopharmaceuticals. So what we are going to learn in this particular question is that we are going to study what are the ICH guidelines which were applicable for biopharmaceuticals, right? Let's see. In continuation of this question, in developing a monoclonal antibody in accordance with ICH-S6 guideline, we have to remember ICH-S6 guideline is a guideline which is used to which mentions about how to what are the studies requirement for biopharmaceutical submission to the regulatory agency. What is the first consideration when designing IND enabling in biosafety studies? So let's see what options we have. Option A, we have selecting a dose level, producing clear toxicity. Option B, we have assuring the test article is non-immunogenic. Option C, we have understanding the mode of action. And option D, we have choosing a relevant test species. As you can see, the right answer will be in the green text, which is answer D, choosing a relevant test species. So for biopharmaceuticals, as per ICH-S6 guideline, choosing a relevant species is the most important criteria, okay, for designing IND enabling studies. So first, what are the preclinical safety testing should consider when you're doing for biopharmaceutical? One is selection of the relevant species. Here, the meaning of relevant species is that, see, when you do a conventional traditional molecules are there, no, you will be doing from mice, rat, rabbit, dog, monkey, like that you will be doing. But when it comes to monoclonal antibodies or biopharmaceuticals, there will be selected species will be there, because the monoclonal antibodies or protein molecules, whatever are there, no, biotechnology derived products, to act on that particular population, on the testing population might be either mice, rat, rabbit, or dog. They should also have that enzyme or the receptor to which this monoclonal antibody can go and bind. Getting my point? So instead of doing studies in a battery of animal lower to higher species, you will be doing in a selective species. So this is the most important criteria. Actually, this is a basic knowledge question actually. But still, there are certain people who might not be working in biotechnology industry, so for that, it is they have to learn it from the beginning. So another important criteria is age of the testing population, physiological state of the test item, I believe, the manner of the delivery, including dose, route of administration, treatment regimen. Okay, whether it is given once in a month or twice in a month, by what route of administration. Stability of the test material is important, because these protein molecules or biotechnology derived products, no, they are subjected to pH or stability related. There will be degradation or the chipping of the what do you say polymer units will be there. Protein therapeutic, if they break, because these are large molecules, large protein molecules made up of small functional units. If because of the excessive pH conditions, if they the small, one or two functional protein molecules they break off, means they will be subjected to degradation. That is what I'm saying, that stability of the test material under the conditions of use in that order, that is very important. And for this reason, for this particular question, please go back and read ICH S6 guideline in detail. This S6 guideline contains preclinical safety evaluation of biotechnology derived products, okay, derived pharmaceuticals, okay. Specifically, they have given that you go back and check in section 1.2 of para 2. Then this question is taken from American Board of Toxicology. As I said, no, they have a 50 series of questions is there. From that question only, this particular thing. Let's in this particular video, we're also going to second question, question number 2. What IND enabling preclinical studies are recommended in ICH S6 as well as ICH M3 guidelines? So this question is mixing two guidelines. One is ICH-S6 as well as ICH-M3. M3 guideline is for conventional molecules. Okay, the battery of typical toxicology studies. ICH-S6 is for biotechnology derived products. So, in by mixing these two guidelines, they're asking in these both the guidelines for both monoclonal antibody and small molecule pharmaceuticals. What are the IND? Or what are the common studies for both type of guidelines it is taken? So you see, reading one single guideline is also not enough. You have to integrate. That is what I keep saying, that whenever you are preparing for any competitive exam, the examiner will mix the same topic. Integration of that questions will also takes place, where it is a brainstorming for you people. Okay, mere reading is not enough. You have to integrate. That is what you have to remember. So we have option number A, immunogenicity studies. Option number B, we have tissue cross reactivity studies. Option number C, we have pharmacokinetic and toxicokinetic studies. And we have option number D, in vitro genotoxicity studies. As you can see, the right answer is in green text. That is option number C, pharmacokinetic and toxicokinetic studies. Okay, so this PK and TK studies are both applicable as per ICH S6 as well as ICH M3. Let's see what is the explanation given by American Board of Toxicology. First one is single and multiple dose PK, TK as well as tissue distribution studies in relevant species are recommended for both type of programs. Okay, then it says immunogenicity, tissue cross reactivity, and unusual for small molecules. For small therapeutic molecules, immunogenicity, tissue cross reactivity are only requirement as per ICH S6. Okay, genotoxin studies for biotechnology derived products, there is no need of doing any genotox studies, right? This is what the important thing. So this is so I have taken it from one of the presentation from one of our seniors in the toxicology industry. So same slides I have kept. So this is what we are discussing about TNF alpha. TNF alpha. This is the you can see it is in human. How the shape changes in monkey, in murine, in dog and bird, mini pigs, and this is the epitope in the molecule. So in order to this epitope to bind in the molecule, only human TNF alpha can bind. Because this is what I am saying. The compatibility between the receptor as well as therapeutic molecule. Okay. Relevant species one in which biological product is active. In this case, this one. Due to the presence of receptor epitope. Right. The next one is binding specificity. As an affinity should be there. There is a lock and key mechanism should be there. And affinity of biological often limits species cross reactivity. Now you can see that this cannot bind with this one, this one, this one. So neither monkey, neither murine, neither dog or mini pig are reacting with this particular epitope in the molecule. Presence of expression, homology, distribution of the target at the site of action in the preclinical species. Even the presence of these epitopes are also there, means they have to be present in what particular part of the body. Site of action in the preclinical species is also important. Distribution of the target is also very important. Then next we have binding to the target or affinity, and PD, pharmacodynamic response in vitro and in vivo potency. Whatever the in vitro results are there, it has to be translocated to the in vivo potency. Should be measurable. We should be able to measure the IC50, EC50, those parameters. The need to conduct a toxicology studies for biologics in a pharmacological relevant model can result in toxicology studies being conducted in single species. Getting my point? The same relevant species you have to do pharmacology studies, as well as in the same relevant species you have to do toxicology studies. In one single species is enough. No need of doing it a battery of toxicology studies. Studies in non-relevant models can be misleading and discourage. See, you have selected a non-relevant model or that particular, for example, you are doing one dog study, but dog is not relevant species and dog is not responding to your drug. It doesn't mean your drug is failing. It means that the drug is not responding in that particular species, but it might respond in monkey. Okay, monkey might have that particular receptor. Getting my point. Surrogate molecule may be useful in defining pharmacodynamic but not recommended for safety transgenic models or corresponding surrogate biological entities for a particular animal may be required in the absence of suitable species. Whatever they are saying is that in case we don't have any species like mice, rat, rabbit, or dog, monkey, any of these species are not responding to our drug, in such cases you can go for surrogate data or else it is not recommended. That is what it is trying to say. I know the topic might be little bit in deep, but you can pause and you can read. These are taken from one of my seniors, as I said. No, in contrast, toxicology studies for small molecules are performed in two species, one rodent and one rodent. Generally, we do it in rat and rabbit, or rat and higher species like dog or monkey. They will be doing case by case based on the chemical. And next we have preclinical safety testing in monoclonal antibodies. The primary objectives of these non-clinical studies are to define pharmacological and toxicological effects of investigational products prior to initiation of the human study. Before initiating clinical trials, you have to do preclinical studies, which includes functional characterization of the product, such as its ability to reduce pathogen load, okay, whatever the pathogen load is there, impair the function of the toxin, promote pathogen clearance from your body, from the blood circulation and tissues, improve clinical signs, reduce weight loss, or reduce severity of the infection. Second important point is identification of the possible toxicity and likelihood of the potential adverse events or undesirable effects. Identification of the safe starting dose. Either for your first in human trials, you have to identify either based on novel approach or novel approach. You have to decide that what is a safe starting dose for the phase 1 clinical studies and dose escalation studies. SAD/MAD studies के लिए single ascending dose, multiple ascending dose studies के लिए. Okay, and PK/TK are undertaken in order to understand exposure in safety studies to allow cross species comparison, okay, between mice and rat or human or rat like that, and to predict margin of safety. MOS, margin of safety establishment, is very very important for clinical trials. Based on that only you will be comparing HED, MHRD, maximum human recommended dose or maximum recommended safe dose. Like this comparison will be there. PK/TK study design and interpretation of the data should consider nature of the monoclonal antibody or the conjugate, its stability, ability bind serum proteins, the presence or otherwise the infection or target antigen expression, and its level in the recipient animal model, as well as the route of administration. So these are the important factors when you're doing PK/TK study design. So this was one of our option. No, this is the option actually. Then we have preclinical safety testing in monoclonal antibodies. Selecting a pharmacological relevant animal species. Throughout the video, we have been discussing the same point. For testing and safety and toxicity of novel monoclonal antibody therapies to support clinical testing can be challenging. Frequently, species of choice is preferred. Generally, monkeys are the choice for the monoclonal antibodies. With the increasing number of maps in the pharmaceutical pipeline, if you see whatever the new drugs which are coming into the market, all the anti-cancer drugs, most of them are the origin is monoclonal antibody, biosimilars. Okay, this has significant implication on the primate use. Okay, that is the reason it is causing ethical concerns as well as economical issues, because doing a monkey study, it will end up with the crores of population, crores of money, I mean to say. Then generally, primate is the only species in which target antigen show cross reactivity with monoclonal antibody. Then this will be the species used in preclinical developing of safety testing. Then we have what are the factors that drive the choice of species, influence the demand for primates in testing the safety and toxicity of monoclonal antibodies include. Number one is expression of the target antigen. The target antigen is much more expressed in the case of monkey. Recognition of the antigen by the monoclonal antibody with sufficient affinity to allow evaluation of the pharmacologic activity. The epitope as well as this receptor, they should show affinity. Knowledge of the relative potency. At low dose, it has to show high effect of the monoclonal antibody in human and animals. Pharmacological relevance. That is, the antigen has a similar role to that in the human, right? Then appropriate tissue expression profile, then immunogenicity and production of neutralizing antibodies, which can make it difficult to achieve high enough levels of the monoclonal antibodies to assess efficacy and toxicity. Okay, so the important conclusion what we have is that therapeutic antibodies have become the predominant class of new drugs developed in recent years. As I said, no, all if you see trastuzumab, edalimumab, or tenecteplase, denosumab, all these drugs are there. MABs which are used for anti-cancer for cancer treatment in the second stage or third stage of cancer treatment are there. No, the new drugs, no, pharmaceutical companies are also going, instead of going for conventional way of traditional drugs, they are choosing to develop in their pipeline. When they do IND investigation new drugs, they are going for biosimilars, they are going for biotechnology derived products, right? Because these no these type of drugs will have less adverse effects because they are going and acting on a specific site, specific receptor they're acting. There will be no unwanted adverse effects will be not be there, which will be having, for example, conventional antihypertensive anti-diabetic drugs. If you see, they used to have lot of adverse effects. They might have some efficacy, but they were affecting your either in the kidney or liver. Such type of drug induced other toxicology adverse effects were there. But In case of monoclonal antibodies or biotechnology derived biosimilar products, no, there is less number of adverse effects are there. So the first point is that one. Complexity of monoclonal antibodies dictates that one cannot prescribe a pathway for the development of all therapeutic antibodies. Hence, it has to be a case by case development pathway. But if you see a conventional molecule, no, there is a well defined based on historical evidence. There is a well defined. First, you will do genotox study, QSAR will be there. Then you will be doing MTD studies. You will be doing. Then you will be doing 28-day or dose range finding studies, 14 days. Like that, there will be a fixed pathway is there because hundreds of traditional molecules we have already studied based on which the IND pathway is defined, well defined. But when it comes to monoclonal antibody, there is once you might go for step one, directly you will go to step three, again you might go to back to step two. So like that, that is what it is saying. Case by case, you have to deal with the development pathway of this type of drugs. The need for constant update of knowledge accompanied with with experience in development of antibodies is essential. That's why whenever the scientists are working on this monoclonal antibodies, or if person who is working in biotechnology industry, no, they have to be well experienced them, because the experience only teaches. Because in the guidelines only 10 12 sentences, 10 pages will be there. But how to handle every type of chemical, that will be learning on job training only you will be getting. Then initial in vitro based mechanistic testing must be following with relevant species. Usually primate toxicity testing is essential. So from this question, what we have learned is ICH S6 guideline you have learned. Then you have learned about ICH M3 also. So what are the studies required for a traditional molecule, conventional molecule? What are the studies required for a biotechnology derived molecule? Okay, and mostly what we are learning is immunogenicity study, tissue cross reactivity study, or what is the relevant species? Mostly monkeys are the relevant species for ICHSC guideline. Okay, hope you are able to learn from this particular video. Thank you. Be curious, and for any questions, please write down a mail to this particular mail ID, and like, subscribe, and follow my YouTube channel Toxicology on Board. All the best for your exam. Thank you.